So our speaker for tonight is Aaron Veale. I'm not sure. Aaron was born and raised in Kamloops, BC. He attended the Thompson Rivers University and earned a Bachelor of Science. As part of his honors thesis, he joined an ornithological lab and looked at the differences in body size between island and mainland dwelling birds. That might be an interesting topic for another time. This led him to his supervisor in Lethbridge, where he is currently doing a project for his master's degree. His undergraduate work also introduced him to the wonderful world of birding. And when he's not in the lab, he can usually be found exploring the outdoors for the amazing birds of Western North America. And I'm particularly, hopefully, the bluebirds are going to be back soon. So I thought this was very topical right now to have a talk about our bluebirds, which are often our first birds back.
Well, on that topic, yeah, as Anne was saying, the bluebirds will be back soon. There's actually, if you follow eBird there and get the rare bird reports, there were actually a couple cited in Medicine Hat not too long ago, and they stuck around for about a week. I'm not sure where they're at now, but that's definitely a good sign. Okay, so thank you for that lovely introduction. So my name is Aaron Veale. I am a second-year master's student at the University of Lethbridge here. I am currently working with Dr. Teresa Berg, as Anne said, at the University of Lethbridge. And then my co-supervisor, Dr. Matthew Rudink, is at Thompson Rivers University back in Kamloops, BC there. So I'll be talking to you today about investigating potential introgression between two North American passerines or songbirds, and I'll explain what's going on with that project in a moment here. But before I get into that, I'll give you a little bit of background just on population genetics and hybridization in general. So our lab studies population genetics at its very core. So this is the idea that we look at the distribution of genes, which are the units of genetic inheritance, and the distribution of the genotypes, which are the genetic sequences that make up these genes. We compare populations to do this. So we compare populations of the same species, or we do this for different species, and we look for differences in their genomes. And if we find any differences, we look for an evolutionary cause to explain why we're seeing them in the first place. So maybe there's an element that's being acted upon by natural selection in one area versus another, or maybe there's just some genetic drift there going on. And a perfect example of this is something like the Steller's jay. Now, being so close to the mountains there, you may see these fairly often. The Steller's jay here can come in a multitude of what we call phenotypes, which are different forms, and it all depends on where you are. Now, in the interior of the northern portion of its range, which is probably the phenotype you're most familiar with, a Steller's jay will have the distinct black crest. It might have a partial white eyebrow, blue streaks on the crown, and a black back or a sooty back there. But if you compare a Steller's jay, so same species, to something like South Mexico or Central America here, you'll see potentially a starkly different bird. So you'll see something that lacks the crown potentially, or the black crest there as much, a bluer back to it, and a more pronounced eye ring. So again, same species. These are technically subspecies, but different breeding populations. So there's something going on there. And it's something that's created genetic difference. So we have to ask ourselves, what creates this genetic difference in the first place? Well, it's all a story about gene flow. So a gene flow is the movement of genes between and amongst populations of a species. And this can be facilitated either actively or passively by the movement of the animals or plants themselves, or their gametes or seeds. So usually we're thinking in terms of physical barriers to start off with. So these physical barriers can come in many forms, one of which is a geographic barrier. So again, in Calgary there, you guys are nice and close to the Rocky Mountains, which for North America is one of the major geographic barriers separating the East and West. Other physical barriers may include oceans or large bodies of water, valleys, like inhospitable areas such as deserts. It's just anything that separates two populations there. Another one of these, again, we have an example right here in Alberta, is a sky island. Now, a sky island is a raised, like a higher elevation habitat that does not reflect the habitat surrounding. So the one in Alberta here is the Cypress Hills, which connect over into Saskatchewan there. And if you take a look, you're driving through, you'll see grassland and kind of this badland habitat. And then suddenly, as you start going up the incline, you'll suddenly find yourself in lodgepole pine forest, fir forest, and trembling aspen, which is quite interesting compared to the surrounding area. And you might be wondering, well, how does this affect birds? Because of course, birds can fly. They're fairly mobile, so raise in elevation really wouldn't cause much, right? But if we look at something a little closer to home, we can look at something, well, down in Idaho there. There is a red crossbill lookalike that was actually found to be its own species. I think it's endemic to what's called the Albion Mountains there in Idaho, and it's called the Cassia crossbill. So this is a crossbill that looks almost identical to the red crossbill, only it has a different beak morphology, and it has a different song type and feeding behavior. So whereas red crossbills are nomadic, they're constantly moving around where the food is, the Cassia crossbill was able to stay in place and just focus on the area that is Sky Island affected. So what's right down in southern Idaho here. And it's even been suggested that the Cypress Hills had their own endemic crossbill species not too long ago. So again, just marked on our map here, we see that according to studies, there was at least a subspecies, it may have even been species level, that was endemic to the Cypress Hills until the American red squirrel was introduced in the 1950s and started competing for the same lodgepole pine cone crop that these birds relied on to remain in the park year round. So now you can find red crossbills in the park, but they're likely just nomadic and not this resident population that we used to find. So back to the genetic differences, there could be something like a fragmented habitat, and this would really affect a species if it has a high fidelity to a habitat type. So if we stick with the crossbills there, they might see a decrease in reproductive success, or maybe they just outright die in an area where they see suboptimal habitat. So if they're not in a pine forest, maybe they start to decline. Or it's something else, maybe they're closely connected to water, maybe a species relies on plant cover, not for food, but for reproductive success. And outside of that given area, it sees a sharp decline in its fitness. And then we also have to consider something called isolation by distance. And this is the given fact that if a population is so widespread and it has what we call disjunct populations, which are unconnected populations, a bird, let's say on the West Coast, will never have an opportunity to connect and mix genes with a bird on the East Coast. So just by the grace of distance, these two groups can become isolated. And we often saw this in cases when we consider the glaciation. Now, one of the major shaping events of North America was the glaciation within the last 20,000 years. And oftentimes we'd have species hiding out on these ice-free areas called glacial refugia. And depending on how the species interacted and how it spread out from there, and how many refugia it was able to colonize in the first place, we can often see different breeding populations doing different things. So right now, it's kind of weird to think about, but we're still kind of seeing the effects of that glacial refugia and that glaciation within the last few thousand years. So again, this is something just to represent the same species, but maybe they have distinct genes that can be used to isolate and maybe characterize their population, their breeding population. So these are all effectively the same species we're saying in this example here. And what we see is they stay within their own kind of confines. We don't see genes that can be used to characterize one population showing up in another. They're kind of isolated to a certain area. And then we kind of cross this with we compare this to another idea, which is kind of the null hypothesis here, is the idea that the genes are being spread around. The gene pool encompasses the entire range of the organism, and mating is essentially random. So genes can be shared with different individuals wherever they are in the range, and it doesn't matter. So in one side, we say that the gene or the breeding populations are structured. And in the other, we say that they're panmictic. There's no rhyme or reason to it, and individuals on one side will have the same genetic composition of the other. And that's something like, I believe, snowy owls have been said to be panmictic, red-breasted nuthatches are said to be panmictic, and a number of other birds too. And if this persists long enough, in the case of a structured breeding habitat, at least, or a breeding population, if it persists long enough, it can lead to what we call allopatric speciation. And that just means that a species gives rise to another species or two different species just based on being in a different habitat. So maybe our bluebirds become a different species here compared to our purple birds over in Ontario, just on the grace that they're in different habitats and geologically isolated. And we often see this if we look at a west versus east split. We have something like our western spotted towhee versus the eastern towhee, the same genus, of course. If we go back to our Steller's jay and we compare that to its equivalent in the east there, the blue jay, which I should note that the blue jay has since done very well to spread up and a little bit past the Rockies, so it's kind of quickly closing that gap. And then we have something like the western meadowlark versus the eastern meadowlark, which again has been a little bit complicated since the eastern has just been recognized to split into the east, and I think it's Miriam's meadowlark now, down in the extreme southeast of its range. But then we also have to have something that maintains the speciation. So once we remove these physical barriers, like we've done with the glaciers, deglaciation has happened in North America so that most of the continent is no longer covered. We have to have something that maintains this original event that gave rise to these different species. So that could be behavioral. So it could be different feeding habits. Maybe it's niche partitioning where one takes advantage of a certain niche where another doesn't. Or it could be maybe something like temporally. Maybe we have a diurnal species versus a nocturnal closely related species, something like that. Something where they don't interact too much, but still in the same environment. It could be based on sexual selection. So it could be a visual display like our peacock there, or it could be a song like something like spotted towhee. And it's something that's fairly exclusionary. So you might have heard that birds across a range exhibit different dialects. And there's actually been studies that show that different dialects are preferred by that regional bird. So if I go back to my hometown in British Columbia there, the spotted towhees will sing differently than they do here in Lethbridge. And the Lethbridge spotted towhees will prefer the ones from Lethbridge compared to the ones from Kamloops. And then there's also this idea of migration behavior. So just because a number of birds maybe winter together doesn't mean they're always breeding together. So here, let's say they went from a central wintering ground on the coast, and now they've dispersed to their breeding grounds. Even though they're intermixing on the coast there, if these two species don't interbreed with each other, then they'll have separation in their genetics, which is quite interesting. So what does that mean in terms of hybrid? Well, sometimes even these secondary or these behavioral barriers break down, and that can lead to something like in the case of secondary contact. Now, if they don't break down, they stay separate species, that's totally fine. That happens a lot of the time where two closely related species occupy similar areas and they never interbreed. As far as we can tell, that's something like your Empidonax flycatchers that seem to really respect those species boundaries. And good for them. Not very interesting though for us. Then you could have something that's not necessarily mutually exclusive of the following orders here, but it's something like introgression. And this is where part of the genome of one species gets incorporated into the other. And I say introgression is not mutually exclusive of the following outcomes that I'll talk about in a moment here, because introgression could be, it arises through hybridization, and it could be ongoing hybridization between two species, or it could be through historic hybridization. It's just anything that kind of leaves a mark that you can find on the genome of the species you're trying to study. So if we take an example here, let's switch it up from birds for a moment and go to, let's say, grizzly bears and polar bears. And of course, we know that there have been cases of where grizzly bears and polar bears reproduce to form a hybrid, a grizzly bear. Now, our first generation hybrid is referred to often as an F1 hybrid. And just based on math and the laws of inheritance, this individual will have 50% grizzly, 50% polar bear, which I realize I've switched the colors there. Now, sometimes these hybrids themselves might not be viable, so they might die before they reach maturity, or they may not be fertile. So they reach maturity, but they can't sexually reproduce. And therefore, they can't continue on the cycle. But in cases of like something like a grizzly bear, it's been shown that it can reproduce. So it might be in an area where polar bears are rare and grizzly bears are more common. So its next mate, the F1, might mate with another grizzly bear. And this would further dilute the F2, or the second generation hybrid, from 50-50 to 75-25. And again, in cases of numerical imbalance, this can go on and on and on. We call this backcrossing. So this is where the hybrid is mating with one of the parental species or both parental species, but usually it's one more than the other. And eventually it can lead to a genome where we only find a very small percentage of that original polar bear DNA. And for all intents and purposes, our grizzly bear is reflective of a grizzly bear there. Sometimes this can be either beneficial, but oftentimes hybrids are acted against. So natural selection, for whatever reason, of course, it doesn't act with reason, but natural selection will pick an element there that it acts against and remove our hybrids from the breeding population. Sometimes this is human-based. I think there was a report not too long ago of a hunter shooting a grizzly bear, which was kind of sad to hear. And then, of course, it's more complicated in the natural world than just two hybrid species, or two species contributing to genetic material there. So back in 2018, there was a paper put out by Toes et al., and it was talking about an F2 hybrid, but its genetics was quite interesting based on what they thought it was. So it was based on an original blue-winged warbler and a golden-winged warbler hybrid. And this hybrid is not common, but it's also not totally uncommon. So it's not uncommon enough that they have a name for it, which is Brewster's warbler. And then this Brewster's warbler went on to breed, again, outside of its species, with a third group, the chestnut-sided warbler. And the chestnut-sided warbler gave rise to this new hybrid that was the point of this 2018 paper, where they tried to figure out what the genetic background of this is. So again, two species, it's not always as easy as that. Sometimes you have output or input from different species all over the place. So if we get back to our comparison, we could also have the invasion of a species. And again, an example of that, and I'll apologize because tonight I'll use a lot of examples. An example of that was seen in 2020 when we actually lost a crow species, but it didn't go extinct or anything. Through a study by Slager et al., they actually found that the northwestern crow, which was native to, thought to be native to the western coast of northern North America there, so from Alaska to British Columbia, just to the top of Washington, was actually found to have been pushed out and interbred with by the American crow. So the more common American crow had encroached on the northwestern crow habitat, and over time, they think through centuries and centuries of breeding, had actually pushed out any purebred northwestern crows to the point where you could only truly find a northwestern crow in around Alaska there. Everything else had some degree of introgression to it. And they have since moved the northwestern crow to a subspecies of the American crow, but that's kind of problematic. We can talk about that after. The other option, they might form a hybrid zone. So at one time, these crows probably formed a hybrid zone, but we saw an imbalance where the American crow was able to invade. A hybrid zone can also take the tact where both parental species can't really gain that foothold, and wherever they meet in commonality in a location, they produce a kind of medium phenotype. We see an individual that has both characteristics of both parent species. So a common intergrade zone, I should say, for this case would be something like a northern flicker, which you've probably seen just at a bird feeder or in your yard. And on the west coast there, we have red-shafted northern flickers on the eastern half of North America, we have the yellow-shafted northern flicker. And wherever they meet, they can often get these integrades within a very thought to be set zone with a mixture of characteristics. There's also another breeding hybrid zone in and around the, again, Vancouver, kind of Washington, northwestern Washington area, where we see Olympic gulls. And these gulls are thought to outnumber their parental species, which is the glaucous-winged and the western gull, at least in this area, just where they meet up. So again, depending on where the hybrid zone is and what type it is, it can either be rigid like this, or it can be something called ephemeral, which we'll circle back to in a bit. Another option here, they could merge back into one species. And this is not to say that they merge back into the same species that they were before the speciation event. Of course, temporally, that species is done. That one has split off. But they could merge back into a single species that now encompasses the genomes of both. And then probably one of the rarest outcomes, at least for animals, they could form a new hybrid species altogether. And of course, I say this is rare for animals because for sexually reproducing animals, you at least need a stable hybrid zone where the hybrids are kind of pushing out the parents and you don't have backcrossing going on. So this can be very rare. Oftentimes hybrids occupy the same niche and they often compete against their parental species. So this kind of eliminates them right off the bat, or kind of puts them back into the fold, if you will, through backcrossing. But for plants, it's a little bit more common. And of course, plants have the ability to asexually reproduce in some cases. Sometimes they're able to exploit a niche that neither parent can. And sometimes they just get lucky with the ability of seeds being placed in an area where neither parent can get to anyways. So now I'll switch gears and I'll talk about the actual study species that we're focusing on. So for my study, like I said, we're looking at two passerines, two songbirds here, and we're looking at two sexually dimorphic songbirds. So here I've just pictured the two males just because it's easier to tell them apart versus the females of the both species. So on top we have the Western Bluebird there, or Sialia mexicana. And from now on, I'll be referring to it in text as Weeble just by its alpha code. And on the bottom, we have the Mountain Bluebird, so Sialia currucoides. And again, the alpha code for that will be MOBL. So these are two species within the genus Sialia. There's only three to begin with the inclusion of the Eastern Bluebird anyways. But all of them are open woodland habitat. They prefer trees, but sparsely populated trees. And oftentimes this is because they are an obligate secondary cavity nester. So that means they need a tree cavity to raise their brood in. Now, in the last century or so, we've often seen bluebirds, all three species, in fact, now, kind of giving up natural tree cavities in favor of nest boxes. And this makes a lot of sense, given the availability of nest boxes, far outnumbering the natural tree cavities that can be created by things like woodpeckers and other natural forces. And the fact that they're often placed in fields with an abundance of insects. And both of these birds are ground-based insectivores in the summer. So they'll raise their brood off of things like grasshoppers, beetles, grubs, and caterpillar larvae. And then in the winter, they'll switch to things like juniper berries and other plant matter. Now, the genus Sialia that both of these birds belong to, or bluebirds as we'd like to call them, originally are thought to have arisen in the Pliocene, so the mid-Pliocene, 3.3 million years ago. If we were to create a family tree, as actually somebody did this for bluebirds, bluebirds compared to their closest relatives, which are other thrushes and American robins that you were talking about earlier, we actually see that bluebirds are fairly low on the family tree, which means they're what we call basal. And they have less, I don't want to say advanced characteristics, but they have less derived characteristics, like a special syrinx that the robins have or other thrushes have, per se. But they're still fairly new on the scene at 3.3 million years. Now, other sources have tried to place when the split is in the species, but it's kind of hard to determine. I can't really see a clear date, but usually it's placed between 3 million to 2.5 million years ago. And I should mention that with using genetic analysis, it was originally thought that just based on morphology, the Western Bluebird and the Eastern Bluebird were the closest related, and the Mountain Bluebird was kind of on the outside of the three. But through genetic analysis that Click and al. Did in 2005, they've actually found that the Eastern Bluebird is closest related to the Mountain Bluebird, and that it was the Western Bluebird that was the first to diverge of the three a number of years ago. Now, when we look at habitat, as the name suggests, the Western Bluebird inhabits most of Western North America. And for our study, we were really interested in the breeding populations here in Western Alberta. There have been breeding populations noted in the east, but we didn't give too much credence to this just because we think it was probably a one-off or a misidentification at the time. But this Western breeding population seemed to be more hypothetical, more well-supported, just based on the fact that we had a established breeding population in Southeast BC here, right across the Rockies. So we could be seeing some reinforcement there. Now you compare this to the more ubiquitous Mountain Bluebird, and we can see its range stretches all the way from Alaska east to central Manitoba, and then its breeding population goes all the way into New Mexico here and Arizona. So it's a little bit more northern of a bird. And this kind of reflects it's said to be more of a colonizing bird. So it has a better time finding a new habitat and then kind of exploiting that. And it's also said to have a higher altitudinal gradient, which kind of gives it its name, the Mountain Bluebird. And this did not go unnoticed. So within the last, well, let's say 80 or so years, we've seen a rapid expansion of the Western Bluebirds from the true West here and kind of moving into the center of the continent. And it was noted back, I think in 2008 here, that this expansion had taken place in the first place. And Duckworth and Semenov, which are two researchers down just across the border in Montana there, were able to find that over the last number of years, this wave, this expansion in the Western Bluebird habitat was actually being led by aggressive male Western Bluebirds. And it had since brought them into areas that for a long time was just Mountain Bluebird habitat. So they were observing, and I think they took data from about 2,500 individuals, and they were finding only at the front of this expansion here, a odd number of hybrids. So what they did an analysis where they essentially took the proportion, came up with something called a Q value, which is the proportion of ancestry, roughly, that can be attributed to an individual. And they came up with a metric for it. They used a histogram bar here, and they denoted it as even being pure or hybrid. So for pure, a bird would look like something between greater than 80% or at 100% there. So let's say the red is Western and the blue is Mountain. So these would be pure individuals. The F1 hybrid that we talked about earlier would be something that's like a 50-50 split. And then they called a late stage hybrid, which would be something like your F2 or F3 hybrid even, as being greater than 20%, but less than that 80% mark. And they found a number of these individuals, like I said. And they came up with this question, why is this happening in the first place? And it all came down to something called Hupp's principle, or the desperation hypothesis. And if you remember back, I just said that Mountain Bluebirds are a better colonizer. So they show up to a spot and they kind of have this low density to begin with, but they're the only bluebird there at the start. As that expanding wave kind of encroaches, they come into contact with the more aggressive male Western Bluebirds. And these aggressive male Western Bluebirds are, of course, migratory. And as a migratory bird, it's a really bad idea to give up a mating opportunity. And if you think about this in just, of course, the birds don't consciously think about this, but if you think about it in the evolutionary terms, to give up a breeding opportunity means you're putting yourself at risk for another year. Migration is a really taxing event, so a lot of birds don't return from it. And you're putting yourself at the risk of not finding a mate next year either. So instead of waiting for the female Western Bluebirds to get their numbers up, the numerically imbalanced aggressive male Western Bluebirds target the female Mountain Bluebirds that are already in higher enough proportions to be targeted in the first place. So we see the Western Bluebird outcompeting the Mountain Bluebird in only a few cases, but enough to cause hybridization here. As time goes on, the Western Bluebird kind of brings its numbers up. That aggressive Western Bluebird front kind of passes, and they're replaced by more docile male Western Bluebirds. And then the Mountain Bluebirds are eventually kind of kicked out. So because it's succession, it's the idea that with the mountains being a better colonizer, they'll eventually move on, and the Westerns will kind of take that habitat over, and the cycle can continue. So this was really interesting to think about. And it also brought up the question, well, what does a hybrid look like in the first place? Can we just identify it out in the field, or does it have to be identified through genetics? And they also answered this to a degree. So here, Duckworth and Semenoff took pictures of the birds they caught in Montana there. And the top row outlined in red, the first two are purebred Western Bluebirds, and the next two are purebred Mountain Bluebirds. And then everything outlined in this black square here was identified as a hybrid in their study. And you can see that the hybrids have a varying degree of both characteristics. So they're not necessarily identifiable in the field just by a cursory glance. You really have to know if there were hybrid pairings in the first place or the genetic backgrounds of the bird. They also got really lucky. And of course, bluebirds are what we call socially monogamous, which means they pair off to raise their offspring, but they're not sexually monogamous. So sometimes a bluebird will have extra pair matings with another close proximately close bluebird in its area. And we actually saw this, again, Duckworth and Semenoff were lucky enough to see this when they saw a hybrid pairing, so a Western male and a Mountain Bluebird female, and the female had an extra pair mating with a purebred mountain male. So this resulted in step siblings, if you will, or half siblings, that they could use to compare these characteristics to. So for my own study, this kind of raised questions in our heads in Alberta here, because there have been sightings through citizen science that have recorded Western Bluebirds in Alberta, and we were kind of wondering if they were doing something similar. So we came up with 3 objectives for our project. The first was to establish the genomic characteristics of a purebred Mountain Bluebird and a purebred Western. The next thing we wanted to do was determine the frequency and distribution of hybrid offspring, if there was any to begin with, in the Albertan breeding populations on their own. The next thing we wanted to do was we wanted to assess for pre-existing population structures. So if you think back to those diagrams with the birds across Canada there, and we wanted to see if Mountain Bluebirds had this structure, or if they were pannyctic, and determine if suspected hybrids had any impact on the genetic makeup of any structure we could find. Oh, I seem to have froze here. There we are. So to do this, we broke it into 2 phases, if you will. So part 1, we wanted to fill the Mountain Bluebird sample gaps. So we did this by going out to eastern Saskatchewan and eastern Alberta there, where we didn't have many samples to begin with. So we focused on Cypress Hills in Alberta. And part 2, we focused on areas of overlap. So this is an area where within the last few years, a Western Bluebird had been seen in and around the breeding period and had been identified through something like eBird, so we could get an approximate location. We typically focus on areas with high density of Mountain Bluebirds, just because that was the easiest thing to do, with the Mountain Bluebirds being far more common than the Western in Alberta. Now, to catch a bluebird was quite the process. So through Teresa, we were permitted to use mist netting with song playback. So this is your standard mist net setup with 2 poles and a very fine mesh of a net set up usually in a treed area here. And we'd use song playback because it taps into that territorial instinct that a lot of the times male birds have in the breeding season. And if you can play your cards right, you can often get the birds to fly right into the net where you can sample them. So here's an unfortunate bird that got tricked by the net and made its way in. I shouldn't say these are not my pictures, these are just general misnetting. And it does work best in a treed area in small clearings. So if you can get something like a corridor here where the birds can traverse through the trees, that's perfect. Unfortunately, if you remember I talked about earlier, with the abandonment from forest to these nest boxes, we don't often have a lot of foliage when working with the bluebirds. And this actually served to be quite a difficulty, especially because the birds are quite acrobatic. Bluebirds, being insectivores, oftentimes would see the nest coming, or see the nets coming, sorry, and fly either around them or over them, and kind of outsmart us to the best of our ability. So we ended up having to come up with a kind of novel design where we came up with this flying V, and we had to focus it in around the nest box. Now this was on fenced off area in Cypress Hills, so this was usually used for grazing. But we did have mild success using this technique. Once we did finally catch a bird, we quickly checked it for injury. Oh, just not, sorry. Yeah, I didn't realize that had sound, but we quickly checked for injury. We took a small, approximately 50 microliter sample of blood, and then we applied a small cotton ball to stop the bleeding, and then we applied filter paper to get a secondary sample. We then attach a silver leg band as permitted by the banding office. And then we measure the tarsus length, the bill length, and the wing length, just to get some basic biometrics. And then finally, once the bird was not bleeding anymore and we were sure it was fine, it was photographed and released. And the next thing we did was we took molecular markers. So I'll talk about the samples we were able to collect here in a moment, but to do this, we needed molecular markers in the first place. So in many eukaryotic organisms, so these are organisms that have a nucleus, the most common way of measuring genetics is either through the mitochondria, microsatellites, which are non-coding regions of the DNA that are neutral, nuclear markers, so those are areas that are found within the nucleus, and then our favorite, we kind of call this the gold standard, single nucleotide polymorphisms, or SNPs. And these are single letter substitutions, so single base substitutions at a specific site for all the individuals. So here we just have a basic example of what a SNP would look like. Our green bird here has, I believe it's an A, and then the bluebird has a G, and the yellow bird has a T at its particular position. And if you get enough of these and they're within a breeding population, you can use these to characterize where the bird came from. So it is seen as part of next generation sequencing, and using a SNP, we could either sequence part or a whole of the genome. So just a minute here, there we are. So to do this, we had to employ two methods. So the first was RADseq, which is restriction enzyme associated DNA sequencing. And this is taking a look at part of the genome. Now you might be asking yourselves, why would you only want to look at part? And the major advantage here is it's cost effective. So you can increase your number of samples, and this is a really good thing to do, especially when you're looking for hybrids. Now the downside, the obvious one to this, is only samples can be randomly sampled along the genome there, and you may miss important parts. So you see here, we have big gaps that we're kind of missing. And you compare this to whole genome sequencing. This is where the entire genome of the organism can be analyzed, but it's costly. It costs, for I think 125 samples, it's about $7,000, so it's not cheap. And it limits the number of samples you can do ultimately if you're not in a huge lab. So just to get this idea across, I like to say it's like driving. So pretend you're with a driver and you're trying to get enough information visually about the road. So RADseq would be closing your eyes every maybe 100 or so meters, which I don't recommend doing, by the way. And you compare this to whole genome sequencing, which is obviously far better. It would be keeping your eyes open the whole time. So whole genome, you might miss something like this deer here, but you might get the general idea of the trees or the forest that you're in. So I'll talk about preliminary results because we're still going through the data. So for our field season, we were able to capture around 116 mountain bluebirds, or get samples given to us by the Mountain Bluebird Trail Society here in Lethbridge. And out in Saskatchewan, we even caught one very surprised looking eastern bluebird. It was actually part of a pair out there. We did not unfortunately find any Western Bluebirds. The only one that was recorded to have been seen in 2020 was ironically April 1st, which was a good April Fool's joke, but it didn't seem to stick around for us. So that kind of complicated things. And as a result, we said those F1s, those first generation hybrids, appeared unlikely based on morphology. But we said that still needed assessment at the time. And I'd like to also point out, we spent a week in southern Alberta and we spent a week in eastern Saskatchewan here. And unfortunately, just due to, we think, the nest box quality, there was a huge disparity in the number of bluebirds we were able to see. Throughout the week, we were monitoring eBird in Saskatchewan there. We could only find about 12 to 16 birds reported. And we compare this to probably the 70 or so birds we were able to catch in southern Alberta. And this is a stark difference. So this was kind of alarming to see. That's just completely anecdotal, but I wanted to put it in there. We were also able to get additional samples. So of course, the samples here, I'll just say marked in the black circle were the ones that we collected in the field. And everything else was either given to us by a museum from North America, so either the Burke Museum or the Denver Museum. And it really expanded the scope of our study. We were also able to thankfully get Western Bluebird samples from Southeast British Columbia there. And this allowed us to continue on with the comparison. And then as a whole, we also collected more from the museums, which was able to lend to the structure idea that we were trying to get at here. So now I'll talk a little bit about my questions. I'll skip over the first one for now, and I'll talk about determining the frequency and distribution of the hybrid offspring in the Albertan breeding populations. So this is a similar idea to what Duckworth and Semenov did with those histograms, where an individual is represented by a bar here. So this is a number of individuals put together. And we have the mountains on one side and the eastern on the, and the western on the other, sorry. And as we can see, when we say a k equals 2, that just means we're telling it that there should be 2 populations here. We can't see any of that admixture going on, that introgression that we should see. So everyone seems to be 100% either mountain or close to 100% either western, which is quite interesting. That appears to say that there is no hybridization right now going on in this breeding population in Alberta, at least based on that evidence. When we just increase that to tell it 3 populations, this kind of gives us the same result. We see a little uptick in introgression, but that's probably just an artifact of telling it the wrong number of populations. We also see, interestingly, at trying to split the mountain bluebirds here, and we'll get to that in a moment because that kind of answers our third question. And then we also look at something called PCOA. So this is a principal coordinates analysis, and this just measures the genetic distance between individuals. And we can see if we compare our axes, our first axis here usually explains the majority of the difference. And that's exactly what we see. The Western Bluebirds are very different from the mountain, but we don't see any individuals that are halfway in between or oddly in between like we would expect to see in a F1 or F2 hybrid. So again, right now, based on the evidence, it seems to say that there are no Western Mountain Bluebird hybrids, at least in that population. Now this brings us to an interesting idea. So this is recent versus ancient hybridization, and this got us thinking, so that Q value or the ancestry proportion works well for the F1 or late stage hybrids are present, but it doesn't work well if there's ancestral hybridization. And this is where we need something called a D statistic. So we're kind of getting ahead of ourselves here, but just to give a better idea, a D statistic is used for ancient introgression. So this would be targeting when the bluebirds first split from each other. And it's limited to SNPs with only 2 forms. So we're kind of limiting ourselves, but we're kind of getting rid of the background noise. And it gives us a, which is considered ancestral, or a B, which is considered the more advanced, more derived. And then we label the populations with P1 being our first, P2, P3, and so on. And then for this analysis, we actually have to use an outgroup. So this was originally done to check how much modern humans, in the Green et al. Paper, it was actually used to check how much introgression modern humans had based on Neanderthal DNA. And for their outgroup, they used something as distantly related as a chimpanzee, which has been split for about 5 million years from humans. And the idea here is P1 and P2 are descended from recent common ancestors, and P3 split at an earlier point. So we haven't done this yet, but right now what we're thinking of doing is we're using some kind of thrush, like maybe another robin or some other thrush to compare as our outgroup. So this would be like our distantly related chimp. And then we'd be using P3 would be our Western Bluebird because it split off first. And then we compare Mountain Bluebirds that are both close within this expansion, this range expansion here. And then we compare also populations that are outside the range expansion. And if we see introgression within or crossover between P3 and P2, which are the Westerns and the Mountain Bluebirds inside their zone, their expansion here, then that gives us a good idea that there was historic introgression in the past. If we see it between P3 and P1, then we have an idea that introgression actually probably occurred back here when the lineages first split off. So they didn't split off completely, and there was introgression going on at the start there. Now, to assess our third point, we were talking about pre-existing population structure in mountain bluebirds, and to see if this had any or was impacted by hybrids in the genetic makeup. Now, mountain bluebirds, just based on our PCOA again, they appear to be a mix. So this is kind of a chaotic. Bluebirds from everywhere seem to be, mountain bluebirds at least from everywhere, seem to be kind of overlapping, and they don't seem to have any distinct genetics attributed to one place. So that means that if the Western bluebirds are contributing into the mountain bluebird population here, it's unlikely that they would create any distinct variation that wouldn't immediately get swamped out based off of incoming migrants from different areas. So that's kind of interesting to see. So again, that just kind of places it in our recent hybridization, kind of another nail against that in that's coffin. And then this is just a isolation by distance matrix, because if you remember, I talked about isolation can sometimes be, or distance can sometimes be an isolating factor. We don't see that either with mountain bluebirds. So there appears to be no structure, and they appear to be pannyctic overall. And just to drive that home, I've just created a few take-home points here. So hybridization varies wildly depending on the study system, as you can see. But right now, at least based off of what we've collected, there is no evidence of early hybridization apparent in Albertan mountain bluebird breeding populations. Mountain bluebirds also appear to be pannyctic, so there doesn't appear to be any structure there, which means, interestingly enough, that a bird like collected in Nevada would have similar genetics to a bird seen in BC or Alberta here, which is quite interesting, I think. Albertan mountain bluebird populations also appear to be healthy and robust. If we compare that, we can actually take also what we call an effective breeding population, which is how many individuals are actually contributing. So that might be another analysis we do later on. And we'll probably do the same for something like Saskatchewan there, just to see if that decrease was just a bad year or if that's on the decline. And then for future directions, like I said, we're now looking for signs of ancient introgression. So that would be the D statistic I talked about. And we've also since included Eastern Bluebird samples from Southwest Ontario and South Dakota there. And this was done because it was also brought to our attention while we were in Cypress Hills that there have been Eastern Bluebirds seen there. And there was even a report on eBird of an unknown, very Eastern Bluebird looking bird that was thought to be potentially a hybrid. So we'll do the same analysis there as well, just to rule it out. We also sent off around 20 samples for that whole genome sequencing. So we're no longer closing our eyes 100 meters and taking parts of the genome, we're taking the whole thing. And we're also planning on scanning the genome for what we call outliers, which might indicate particular genes that are being acted upon or acted against in mountain bluebird populations. So that would be my talk for today. I'd like to put a special thanks out to my home university of Thompson Rivers there, the University of Lethbridge as well. I'd also like to thank the NSERC organization for funding both my supervisor and as a result me. And I'd also like to thank the Mountain Bluebird Trail Conservation Society here in Lethbridge for all the support they've also given my project. I'd like to thank you all for listening. And if you have any questions, I'll gladly take them now. Otherwise, you can also reach me at that email listed below there. Oh, and I'd like to thank my lab mates and Teresa as well.
Very interesting. So bottom line is you didn't see any evidence of hybrid bluebirds. I remember, I think it was last year, there was some report that there may have been a hybrid, but my memory may be wrong.
Yes. Yeah, I was about to say, yes, I think you're right, because if we're looking at the same one, I think there was one even seen back in 2020 that could have been, oh, absolutely, yeah. Sorry, I'm just seeing the chat there. That could have been identified as possibly a hybrid. But again, unfortunately, we weren't around at the time to get that genetics, that sample.
Right.
But it's interesting to see that just in Montana there, it's probably as far as, if I had to guess, Waterton National Park seems to be that cutoff right now where the eastern or western bluebirds have made it this far east. But they haven't made it really into Alberta yet on a consistent basis, which is quite interesting. That might change in the next few years.
Yeah.
Just depends.
Could you just put your email into the chat?
Yeah, absolutely.
Somebody has asked for your email again.
Yeah.
And then there are some questions. First one says, I believe mountain bluebirds tend to come back to the same area they're born. Would this lead to speciation pressure?
Oh, that's a really good question. Okay, just one moment here. I'm just typing in my email there.
Yeah.
There we are. Yeah. So it is, I was actually trying to find this out not too long ago because I was trying to figure out the same thing. So you're right. If they have high, what we call natal phylopatry, so they return to the same area year after year and where they were born, then you would think that this would isolate and concentrate those genes to a particular region. So this would concentrate over time and lead to a speciation event eventually. But for whatever reason, we say it takes the rule of one. So even just one migrant between populations is enough to kind of stir things around. And if there's a chain going all the way up, let's say from Nevada to, again, the north here, that one or two migrants might be enough to kind of stir things up and keep things from really being clear and structured. So that's quite interesting. Right now, they don't seem to be on their way to speciation, but you never know.
Okay. Another question, do you have access to useful data in that Montana Western Bluebird front, or can you go get samples there?
Oh, I wish. In hindsight, we should have. Unfortunately, it's in academics, it's a little taboo to go into somebody else's like turf, if you will. So
Yeah.
Duckworth and Semenov, I think it's Renee Duckworth, she's been working with the Montana Bluebirds for the past 30 years. So I wouldn't want to step on her turf. But I did reach out early on to see if we could get samples from them. And unfortunately, just for whatever reason, it didn't work out. But I would love to see those samples as well. That would be really, really interesting. Yeah, that'd be a dream.
Okay. And Nina says, why are there so many subspecies of crossbills?
That's a good question. Just because I brought it up, that's a fair point to ask. From my understanding, it has to do with their specificity to their kind of host tree. Apparently, they have a very specific beak morphology, especially related to the cones of that specific tree. So the crossbill subgroup or subspecies that back home, for instance, for me, is the ponderosa pine, because that's the one that they are closely associated to. So it seems to be largely based off of their feeding habits. But again, it might just be based off of feeding habits and song, or just how they play out. It's quite interesting. It's an interesting complex.
Okay, any other questions from anybody? Excellent presentation.
Thank you very much.
I guess everybody's just trying to take in everything there.
Yeah, I know it's a steep learning curve, is what I was told when I first started, and boy, I could say it is.
Okay, so if the Western Bluebird wave continues, could it make the mountain bluebirds endangered?
See, that's an interesting question, because right now we're trying to figure out, they haven't seen it, they haven't reported it, at least in Montana. And that seems to be because the Westerns seem to stop at a certain altitude where they just do not like the environment anymore. So if the Mountain Bluebirds just continue up a mountainside, they can still be found within the same habitat, but just a ways up. Right now, I think, if I had to guess, I think Mountain, it might be Eastern, but Mountain probably has one of the largest numbers to it. It's listed as least concern, so it would have to take a lot to get into that threatened range.
All right, okay.
Yeah.
And so, all right, good. So you have one more year in your master's, or?
Technically, I think it's 6 months now.
Oh, good.
Yeah, it's coming up. It's getting to be crunch time.
Good for you.
Thanks.
Good. Okay, so very thought-provoking, lots to think about and observe.
Yeah. Well, you know, as a birder, I'm always keeping an eye out for hybrids. And I think in Calgary there, you guys have had a number of sightings of numerous hybrid birds there. So that'd be awesome to see too.
There's a lot of the hybrid ducks particularly.
Yeah.
There's another question here. Do Western and Mountain eat different insects?
That's a good question. I don't believe they do, but I'd have to see a study on that to be more specific. Yeah.
Interesting.
Yeah, I think they'd probably target the same insect communities. Yeah.
So thanks everybody for attending, and thank you very much, Aaron, for that great talk. And good luck in your last 6 months.
Thanks, Anne, and thanks everyone for listening.
Okay.
Have a great day.
Take care, everybody. Bye-bye.
See ya.